Think about how you've grown over the years, from little kid to who you are today. Mitosis is the force behind that transformation. Your body constantly generates new cells to support growth, healing, and renewal. Even something as simple as your fingernails growing. Yep, that's mitosis in action. Picture this. Right now, in your body, there's a single cell, just hanging out and minding its own business. But what happens when it decides to divide? Now we have two cells, each ready to take on new tasks. And they multiply? Tons of new cells are being created. This is happening all the time in your body, helping you to heal and grow. Mitosis is the process by which a single cell divides into two identical daughter cells. In our lab experiment, we observed mitosis in garlic root tapes, where we witnessed the fascinating stages of cell division up close. Throughout the video, I'll guide you through each stage of mitosis step by step, exploring how cells replicate their DNA, align their chromosomes, and finally divide into two. Let's embark on this cellular journey together and uncover the mysteries of mitosis. Mitosis is a relatively short phase of cell cycle compared to interphase, which accounts for about 90% of the cell cycle duration. Interphase is the longest phase where the cells prepare for division. It consists of three subphases: G1 phase, S phase, and G2 phase. Mitosis is the actual process of cell division consisting of several stages: prophase, metaphase, anaphase, and telophase. In prophase, the chromatin fibers begin to condense into visible chromosomes, each consisting of two sister chromatids joined at the centromere. The nucleus disappears and the nuclear membrane starts to break down, allowing microtubules to access the chromosomes. Meanwhile, the mitotic spindle made up of microtubules begin to form from the centrosomes, which move toward the opposite poles of the cell. Then comes metaphase, where the chromosome guided by the spindle fibers align along the metaphase plate at the cell center. This precise arrangement ensures that each new cell will receive an identical set of chromosomes. The kinetic cores, protein structures on the centromeres, attach to the spindle fibers, preparing for the chromosome separation. During anaphase, the sister chromatids are pulled apart by the shortening of spindle fibers attached to their kinetochores. Each chromatid, now an individual chromosome, is dragged toward opposite poles of the cell. This movement ensures that both daughter cells will inherit identical genetic material. The cell elongates that non-kinetochore microtubules push against each other. Finally, telophase marks the reformation of two new nuclei. The chromosomes start to decondense back into chromatin, and nuclear envelopes reappear around each set of the chromosomes. The spindle fibers break down and the nuclei reform. The stage is followed by cytokinesis, where the cytoplasm divides, officially splitting the parent cell into two identical daughter cells. Now that we understand the stages of mitosis, let's connect this to our experiment. When observing garlic root tape cells under the microscope, we can identify different mitotic phases based on the chromosome arrangement and nuclear changes.
It's anaphase.
You mind if I check?
Sure.
Anaphase.
Like I said. Metaphase.
This is a memorable scene from the movie Twilight, where Edward and Bella observe the stained garlic root tip cells under their microscopes. To obtain the vampire ability, let's dive deeper into this lab. For this experiment, we'll need the following materials: garlic root tips, 1 mole hydraulic acid, fixative solution, 0.8 gentian violet stain, a compound microscope, microscope slides and cover slips, scissors, forceps, a glass watch, and a dropper. First, we cut about 1 centimeter of actively growing garlic root tips and place them in a glass container filled with fixative solution. This preserves the cells and prepares them for staining. Let them sit for more than 3 minutes. Next, we transfer the root tape to a glass watch and add a few drops of 1 molar hyaluronic acid. The acid softens the tissues, making it easier to spread the cells later. We let them soak for 5 minutes before rinsing them thoroughly with water. Now we add 2 to 3 drops of 0.8% gingival stain to cover the root tips. The stain binds to DNA, making the chromosomes more visible under the microscope. After 3 minutes, we rinse off the excess stain with water. We carefully place 1 stain root tape on clean glass slide and add a drop of distilled water. Using the eraser end of the pencil, we gently press down on the cover slip to squash the tissue. This spreads of the cell into a single layer, allowing us to see mitotic stages clearly. Now we place the slide under the microscope, starting at low power. We locate the root tip cells, then switch to higher magnification to examine the stages of mitosis. We identify interphase, prophase, metaphase, anaphase, and telophase based on chromosome arrangements. To recognize the different stages of mitosis in cells, we can follow these steps. For prophase, look for the visible chromosomes that appear at the thickened coiled structures within the cell. The nuclear membrane begins to break down and the spindle fibers start to form. In metaphase, the chromosomes align in a straight line at the center of the cell, known as the metaphase plate. The spindle fibers attach to the centromeres of the chromosomes. In anaphase, the chromosomes are pulled apart as the sister chromatids move toward opposite poles of the cell. This phase is marked by the separation of the chromatids. Finally, in telophase, the chromosomes reach the opposite poles, and the new nuclear membrane begins to form around each set of chromosomes. The chromosomes start to decondense, returning to a less visible state. As we observe, we count the number of cells in each mitotic stage and record our results. This data helps us analyze duration of each phase in the cell cycle. Studying mitosis in garlic root tips isn't just a lab exercise, it has real-world significance. Scientists use similar staining and microscopy techniques to study cell division in various fields, including cancer research. Since cancer is caused by uncontrolled cell division, observing mitosis helps researchers understand how cells proliferate abnormally. In agriculture, studying mitosis in plants helps improve crop growth and breeding. By analyzing how plant cells divide, scientists can develop more resilient crops with higher yields. I would like to express my sincere gratitude to everyone who made this learning experience possible. A huge thank you to my instructor, Professor Jun Lee, for his dedication to teaching and his unique approach to learning. His splendid teaching system, combining lectures, labs, think pair shares, and ambassador programs, challenged us to think beyond the classroom and actively engage with biology. Beyond his teaching methods, we especially appreciate how he takes the time to remember each of our names and personally engage with every student. His willingness to talk with us individually, answer our questions, and encourage our curiosity made a real difference in our learning experience. A special thanks to DKU for providing the resources and environment that made this project possible. To my classmates and teammates, thank you for your collaboration, discussions, and shared enthusiasm that made this studying more interactive and enjoyable. Lastly, a heartfelt thank you to my families and friends for their support and encouragement.